Chapter 10, Single Subject Research. So far, we have mostly discussed group research methods in which researchers collect and analyze data from a large group of participants. An alternative to this type of research method is called single subject research, where data are collected from a single participant or a small group of them. For instance, Van Hall and his colleagues conducted a study in 1968 on six elementary school students who repeatedly exhibited disruptive behaviors at school. First, they established the baseline. That is, they recorded whether each student was doing homework every 10 seconds during 30-minute period. Next, they introduced a treatment in which the teacher gave students positive attention, such as stating good work or patting their shoulder whenever the student was doing their homework. Researchers found that during the treatment phase, not only did all of the students spend significantly more time on schoolwork, but their disruptive behaviors also decreased. During the baseline, one of the students, Robbie, spent only 25% of his time on schoolwork and was engaged the other 75% snapping rubber bands, playing with toys from his pocket, and talking and laughing with peers. But his behaviors dramatically changed during the treatment phase. He spent 71% of his time on schoolwork and only 29% on other activities. What do you guys think happened when teachers stopped giving positive attention? Students studying decreased and disruptive behavior increased. This confirmed that the positive attention changed students' behaviors. This study was among the first to demonstrate that students' behaviors can be quickly and effectively manipulated by the attention given by teachers in an applied setting. In this chapter, we will focus on studies like this called single subject research. More specifically, we will discuss who conducts this type of study, why they do it, and how the data are analyzed. We will also compare the strengths and weaknesses of single subject research and group research methods. 44, section 44, overall of single subject research. As we have discussed, the single subject research method involves studying a single participant or a small group of participants. This approach is also known as small n designs, where the n represents the number of participants. The single subject research is different from the group research, which compares group means and standard deviations. We have mostly focused on the group research up to this chapter because it is the most common approach. This does not mean the single subject research is not important. In fact, this method is primarily used in applied areas of psychology, such as clinical settings where clinicians test the effectiveness of treatments, and also in educational settings as well. You may recall case studies we previously discussed in this course, and you may think single subject research is the same thing. They are, in fact, not the same because single subject research is quantitative, whereas a case study is a qualitative research method. Single subject research focuses on observing and analyzing objective behaviors in experimental settings. In contrast, researchers in case studies take an in-depth approach to analyze and describe a single or small group of participants to understand their subjective experiences through detailed interviews. There are assumptions for using the single subject research method. The first assumption is that it is important to focus on the behaviors of individual participants rather than group behaviors. For example, if one treatment was effective only for half of the population, but it was harmful for the rest, the overall effect would cancel out in a group research method, resulting in finding no treatment effect. In this case, single subject research would be appropriate. The second assumption is that it is important to focus on finding causality. In single subject research, an independent variable is manipulated to control extraneous variables. Therefore, this research is often considered a type of experimental research with good internal validity. For example, the study conducted by Hall and his colleagues manipulated the treatment to see if it influenced students' behaviors. The third assumption is that it is important to focus on examining effects that are both strong and consistent, especially if these effects have biological or social significance. Applied researchers are often interested in treatments that are meaningful in real-world settings, which is sometimes referred to as social validity. For example, Hall and his colleagues' study demonstrated good social validity because it showed reliable and strong effects of positive teacher attention on a behavior that is clearly important to teachers, parents, and students. Additionally, this intervention was practical as teachers reported that the treatment was easy to implement even in their elementary school classrooms, which are often super loud and chaotic. If you look at the history of psychology, you will notice that early psychologists use single subject research methods. For example, Wilhelm Wundt, who was a physiologist, was among the first to begin thinking about psychology as a science. He also established the world's first experimental psychology lab in Germany in 1879. Hermann Ebbinghaus, a psychologist, was the first to investigate the memory using an experimental method in the late 1800s. Ivan Pavlov, a physiologist, studied reflexes in dogs and classical conditioning in the late 1800s. Back in their time, early researchers in psychology, including Wundt, Ebbinghaus, and Pavlov, utilized single subject research in which they collected data from a small number of research participants, including the animal participants. This is partly because sophisticated statistical methods were not readily available at that time. In his research, B.F. Skinner, a radical behaviorist from the mid 1900s, refined and expanded upon single subject research methods and the underlying assumptions. He, along with other behaviorism researchers, utilized these techniques to illustrate how rewards, punishment, and external factors influence behavior over time. In Skinner's approach, known as the experimental analysis of behavior, most studies use non-human subjects like rats and pigeons. Behaviorism was once widely popular but declined in popularity by the mid 1900s. Nevertheless, behaviorism remains a significant subfield of psychology, still primarily using the single subject research method. By the 1960s, numerous researchers started applying this method to conduct applied research, mainly involving humans, leading to the development of the subfield known as an applied behavior analysis. Today, applied behavior analysis is crucial in modern studies related to developmental disabilities, education, organizational behavior, health, and various other fields. So, most research on a single subject is conducted from a behavioral perspective. However, this method can be applied to questions from various theoretical perspectives, such as evaluating learning and memory in cognitive research or examining the effectiveness of new therapeutic approaches by clinical psychologists. Okay, next section, 45, single subject research designs. So let's take a look at what the single subject research results may look like. In the graph of generic study on the slide, the y-axis shows the dependent variable, which is measured repeatedly over time and is plotted on the x-axis at regular intervals. You also see that the study is divided into three separate phases, A, B, and A. In this generic study, participants initially experienced condition A, or phase A, then moved on to condition B, and finally returned to condition A. It's important to note that in single subject research, the shift between conditions generally depends on the participant's behavior rather than a fixed amount of time or observations. Specifically, researchers do not proceed to the next phase until the participant's behavior stabilizes. This helps reduce the noise in the data, making it easier to identify behavior changes once the condition shifts. This approach is known as the steady state strategy. This ABA design, also known as the reversal design, is the most common basic single subject research design. The first phase, A, is a baseline. During this phase, targeted behaviors are observed without an independent variable to establish the baseline. Therefore, it serves as a control condition. Researchers move on to the next phase, B, once the targeted behaviors stabilize. This second phase, B, the independent variable or the treatment is introduced. The target behaviors should increase or decrease because of the presence of the independent variable. Once the behaviors stabilize, the independent variable is removed in the second baseline phase A. This basic ABA design can also be extended by adding more phases to create the ABAB design or the ABABA design. The study conducted by Hall and his colleagues used an ABAB reversal design. The graph on the slide shows the data for Robbie, one of the students studied by the researchers. The percentage of time spent studying, which is the dependent variable, was relatively low during the first baseline, phase A. During phase B, it increased, but then decreased during phase A and increased again during phase B. The reason to include the reversal or the removal of the treatment or the independent variable in this research was to rule out the possibility that the change of the dependent variable was caused by factors other than the independent variable. If the A-B design is conducted, for example, researchers cannot be confident whether the change in the dependent variable is due to the independent variable or an extraneous variable. In contrast, if the dependent variable changes with the introduction of the independent variable and then changes back with the removal of the independent variable, researchers can be more confident that the independent variable is the cause of the change in the dependent variable. This also increases the internal validity of the study. Another variation of the ABA design allows researchers to test more than one independent variable. This is called an ABC-ACB design. In this multiple treatment reversal design, after the phase A baseline, the first independent variable is introduced in phase B. Then the second independent variable replaces the first independent variable in phase C. Then the phase A baseline is reintroduced, followed by phase C, then phase B. For example, after establishing the phase A baseline, teachers give positive attention to students when they engage in studying in phase B, then switch to a different treatment where mild punishment is given when students are not studying in phase C. Afterward, the condition returns to baseline in phase A, then moves to phase C, where mild punishment is given to students when they are not studying. Finally, the condition reverts to phase B, when which involves the positive attention. In an alternating treatments design, which differs from the ABC-ACB design, two or more treatments are rotated rapidly on a fixed schedule. For example, positive attention might be given one day, followed by mild punishment for not studying the next day, and so on. In another variation, one treatment could be applied in the morning and a different treatment in the afternoon. This design provides a quick and effective way to compare treatments. There are two potential problems with the reversal design, both of which have to do with the removal of the treatment. Imagine that you are the participant receiving a treatment that was actually working, but in the reversal design, researchers would take it away when moving to the next phase. It would be clearly unethical to remove the treatment that is actually effective. Another problem is that the dependent variable may not return to baseline when the treatment is taken away. If the treatment a participant is receiving is effective and has a lasting impact, removing it in the following baseline phase would not have much influence on the dependent variable. But it may also mean that the treatment was not really the cause of the change in the dependent variable in the first place. To resolve these issues, researchers can use a multiple baseline design. There are three different types of multiple baseline designs. The first type of this design is called multiple baseline design across participants. In this design, a baseline is established separately for each of several participants, and the treatment is then introduced for each one. In this design, there are two phases. One is the baseline, and the other is the treatment. Therefore, it is an A-B design. The key feature is that the treatment begins at different times for each participant. If the dependent variable shifts when the treatment is introduced to one participant, it could be coincidental. However, if multiple participants show changes in the dependent variable at different treatment times, it strongly suggests that there is a real effect rather than a coincidence. The second type of this design is called multiple baseline design across behaviors. This design is a variation of the multiple baseline design. It involves setting up separate baselines for the same participant, each corresponding to different dependent variables, with the treatment implemented at different times for each variable. The logic remains the same as before. If the dependent variable changes after a single treatment is introduced, it is difficult to determine if the treatment caused the change. However, if the dependent variable changes when all different treatments are introduced, particularly when the treatments are applied at different times, it becomes much clearer that the treatment is the cause of the changes in the dependent variable. And this is the last type of this design. This is called multiple baseline design across settings. This design is an extension of the basic multiple baseline design. It involves establishing baselines for the same participant across different settings. For instance, a baseline might measure the time a child spends reading during free time at school and at home. Subsequently, treatment such as positive attention is introduced first at school and then at home. If the dependent variable changes following the treatment in each setting, this suggests that treatment is likely responsible for the observed changes, increasing the researcher's confidence. So, as discussed, unlike group research design, single subject research focuses on individual participants. Group research analyzes averaged data across participants, such as means, standard deviations, correlation coefficients, and so on, to identify general patterns, and then uses inferential statistics to determine if the results from the sample can be generalized to the population. In contrast, visual inspection of the graph rather than the statistical analysis, such as inferential statistics, is usually used in single subject research. When visually analyzing single subject research data, it is important to carefully observe changes in the level of the dependent variable from one condition to another. If the dependent variable is significantly higher or lower than in another phase, this indicates that the treatment likely had a meaningful effect. A gradual increase or decrease of the dependent variable across phases may also suggest an effect, especially when a trend changes direction. Generally, if a change in the dependent variable occurs shortly after a change in conditions, it suggests that the treatment was responsible for that change. If you look at the top graph, you notice that the changes of the dependent variable from condition to condition are rather abrupt, a strong indication that the treatment was responsible for the changes in the dependent variable. In contrast, the changes in level in the second graph are minimal between conditions, suggesting that the treatment was not working. Another way to analyze the results of single subject research is to use a statistical procedure that is also used in group research, including calculating means, standard deviations, and inferential statistical tests such as t-test and analysis of variance. Another method provides calculating the percentage of non-overlapping data for each participant. This is called PND. This method, PND, assesses the proportion of responses during treatment that exceed the most extreme response observed in the control condition. For example, if a treatment increases a desirable behavior related to the control condition, a higher PND should be seen, indicating a stronger treatment effect. Nevertheless, in single subject research, statistical methods are generally considered complementary to visual inspection rather than replacements. Okay, this is the last section of this chapter, section 46, the single subject versus group debate. Both single subject research and group research are quantitative approaches trying to establish causal relationships by manipulating an independent variable, influencing the dependent variable while controlling extraneous variables. However, important differences exist between these two approaches, often resulting in disagreements. It is useful to explore the main points of contention between single subject and group researchers, along with ways to resolve them. As we will discuss in this section, these two methods are likely best understood as complementary rather than mutually exclusive. Disagreements primarily concern data analysis methods. Advocates of group research criticize that visual inspection is insufficient for determining treatment effects on a dependent variable, especially when effects are weak or data are noisy. They also point out that visual inspection can be inconsistent, with different researchers arriving at different conclusions. Additionally, it is challenging to summarize or compare results across studies using visual inspection, unlike quantitative measures of relationship strength in group research. Single subject researchers generally share these concerns, but argue that their steady state strategy, along with focusing on strong, consistent effects, reduces these issues. When effects are hard to detect visually, they try to enhance effect strength or minimize data noise by controlling extraneous variables, such as administering treatment more consistently. Many researchers also note that statistical analysis is increasingly used alongside visual inspection, especially for cross-study comparisons. Some advocates of single subject research, on the other hand, raises concerns about how group researchers analyze their data. They argue that relying on group averages can be very misleading. For example, if a treatment produces a strong positive effect on half of participants and an equally strong negative effect on the other half, as a standard between subject experiment might see these effects cancel out. As a result, the treatment group's mean would resemble the control group's mean, suggesting no effect even though every participant experienced a significant impact. Nevertheless, group researchers also recognize the importance of examining the distribution of individual scores. For instance, bimodal distribution in scores, including some participants were positively affected while others were negatively affected, can be identified through histograms. Additionally, within subject designs enable researchers to observe individual effects directly and determine the percentage of participants showing strong, moderate, weak, or negative responses. Finally, factorial designs help explore whether the effect of an independent variable varies across different groups, such as introverts versus extroverts. A common point of disagreement between single subject and group researchers involves external validity, which is the degree to which study results can be applied to larger populations beyond the specific individuals or situations studied. Advocates of group research emphasizes the difficulty of determining if findings from a small sample can be generalized. For instance, in a single subject study where a treatment successfully reduces self-injury in two children with intellectual disabilities, it is still uncertain if the same treatment will be effective for other children with similar delays. Single subject researchers, in fact, do agree with this concern. They point out, however, that the strong and consistent effects they usually focus on, often seen even in small samples, are likely applicable to the larger population. These researchers also emphasize the importance of replicating their findings. When they find an effect in a small group, they generally attempt to replicate it with another small group, possibly with slightly different participants or conditions. Each successful replication increases their confidence in the broad applicability of the results. In addition, single subject researchers often highlight that the principles of classical and operant conditioning, many of which were identified through single subject methods, have been effectively applied across a diverse array of species and contexts. As a counterargument, single subject researchers have raised concerns about the external validity of group research. Again, highlighting that studying large groups does not fully address the challenge of generalizing findings to individuals. For example, a treatment might show a small positive effect on average in a large group. However, within that group, some participants might have experienced a large positive effect, others a small positive, and some even a small negative effect. Therefore, applying this treatment to another large group might result in a small average effect, but individual responses could uh, vary widely, making it impossible to predict whether it will be small, large, or negative for any specific person. Another point that single subject researchers often make is that group researchers encounter a similar challenge when they study one specific situation and they try to apply their findings to other scenarios. For instance, researchers examining the impact of cell phone use on drivers using a computer simulation might aim to extend their conclusions to various real-world driving conditions. However, this involves generalizing from a single case to a broader set of situations, which is more complex than simply increasing the number of participants. Effective generalization depends on carefully considering how similar the participants and situations are to those in the target population. Just as quantitative and qualitative methods complement each other, single subject and group research balance each other's limitations and maximize their strengths when answering different questions. For instance, single subject research is particularly useful for testing treatment effectiveness by examining its consistent impact on an individual, especially in biologically or socially meaningful contexts. In addition, this method is useful when the specific behavior of an individual is the focus of the research, such as in a situation where seeking an option for an individual in clinical or educational settings. These are weaknesses of the group research method, but group research, on the other hand, is ideal for exploring the effectiveness of treatment in a group setting. For example, group research can detect a small treatment effect, which can guide adjustments that ultimately produce a stronger, more significant result. Another advantage of the group research approach is its ability to analyze interactions between treatments and participant characteristics. For example, if a treatment works for highly motivated individuals but not for less motivated ones, a group design can identify this more efficiently than a single subject approach because single subject research involves a smaller sample size. Additionally, when variables cannot be manipulated, like the number of siblings, personality traits, or cultural backgrounds, researchers must use group research. So it should be clear by now that no single research method is superior to others. Each method has its strengths and weaknesses. For example, the true experimental research method has high internal validity, but it usually has low external validity. In contrast, non-experimental research methods such as correlational research have good external validity but poor internal validity. When a specific theory is supported by a variety of research findings, such as experimental research and correlational research, scientists increase their confidence in the theory because they have converging evidence. Imagine you left a chocolate chip cookie on the kitchen table in the morning, but it's gone when you come home in the evening. None of your kids admits to stealing it, but your youngest kid has a chocolate stain around his mouth. even though you do not have any chocolate snacks at home besides your cookie. Your oldest kid said she saw your youngest in the kitchen earlier that day, and when he left, she noticed the cookie was missing. Another kid of yours stated that the cookie was there before your youngest entered the kitchen. Although you do not have proof, these pieces of evidence and logic point to one conclusion. Your youngest kid stole your cookie. You're not 100% certain, but it is very likely according to the evidence you have. In other words, converging evidence suggests that he, the youngest kid, is the cookie thief. Scientists use the same procedure. They usually do not say they have scientific proof, but they say they have scientific evidence. Unfortunately, the media often reports sensational conclusions based on the findings of a single study. Scientists, on the other hand, make decisions based on multiple findings from various research studies with different designs and procedures. Scientists do not rely solely on a perfect experimental research study, but focus on overall trends across many studies, like you would rely on multiple pieces of evidence consistently showing who stole your cookie. The idea of converging evidence encourages us to analyze patterns of flaws across the research literature because these patterns can either support or weaken the conclusions we aim to draw. Note that scientists make decisions not based on opinions. Instead, they increase their confidence only when there is abundant evidence to support a claim, and evidence is the only factor that leads to consensus among them. This is the end of chapter 10.